Alright, Assalamu alaikum everybody. How are you all? Welcome to my YouTube channel Biology by HR. Today we are going to be looking at February March 2022 paper 22, which is of AS level biology, and the paper is of 1 hour and 15 minutes. So let's begin with solving this paper. So I already had this paper solved, so I have attached the PDF of my solved solution. So I am going to guide you guys about the question and the answers. Alright, so let's begin with question number 1 over here. So question number 1 part A over here says that table 1.1 shows 3 of the processes by which substances in the solution can move across the cell membranes. It also lists 5 statements that may apply to each of these 3 procedures. Alright, so basically they have told us about the transport processes in which there is the active transport, facilitated diffusion and the simple diffusion. So in short, in the question they are basically asking us that use a tick to show that the statement applies or a cross to show that the statement does not applies. Alright, so they have already shown the first row for us, that there is the movement of oxygen into the red blood cell by the process of the simple diffusion, and the facilitated diffusion and the active transport processes are not involved. Alright, so before we begin solving this question, we should have some prior knowledge about the transport processes. In the transport processes, the facilitated and the simple diffusion are basically the passive processes and they do not require any energy, whereas active process is an energy. Active transport is basically an energy requiring process. And over here we can also tell one more important thing, that the active transport as well as the facilitated diffusion, they both require the carrier protein, whereas if we talk about the simple diffusion, so it does not require the carrier protein, right? It does not require the carrier protein. So now let's look at the other statement. It occurs in both animal cells and the plant cells. So yes, we know that the active transport, the facilitated diffusion and the simple diffusion, it occurs in all the animal cell as well as the plant cell. And if we guys talk about the use of the carrier protein, so I already told you that the usage of the carrier protein is basically the characteristic of the active transport and the facilitated diffusion. It is not the characteristic of the simple diffusion. Alright, simple diffusion does not require the carrier protein. Then if we talk about the other statement, which is the movement of non-polar molecules between the fatty acid tails of the phospholipid molecules, we know that the fatty acid tails of the phospholipid molecules are non-polar, which means that they are hydrophobic in nature. So the movement of the non-polar molecules, they allow the movement of the non-polar molecules, right? If the tails are non-polar, so they will allow the movement of the non-polar molecules. So the substance can pass through the simple diffusion as well, right? There will be no need of the facilitated diffusion or the active transport. Now in the next statement, basically they are saying that movement of ions down the concentration gradient, right? Down the concentration gradient, first of all you will cross the active transport. Why? Because the movement of ions in the active transport is against the concentration gradient. It is not down the concentration gradient. Down the concentration gradient is either facilitated diffusion or the simple diffusion, right? And in the simple diffusion, the movement of ions cannot take place. Why? Because in the simple diffusion there is no carrier protein used. So ions या फिर polar substances cannot pass via the hydrophobic core of the phospholipid bilayer. So what is basically the hydrophobic core of the phospholipid bilayer? If I draw the phospholipid bilayer over here, so this is basically the hydrophobic core, which is made up of the basically the tails of the phospholipid, right? So the ions which are basically polar in nature cannot pass through the hydrophobic core of the phospholipid bilayer by the simple diffusion. So hence only facilitated diffusion can be used over here. Alright, so I hope that this part of question number 1 is clear to all of you. Now let's move on to part B of this question. Part B of this question says that figure 1.1 is a simplified diagram which is representing a transverse section of a part of a young root. The diagram is not to scale. Part 1 is saying on figure 1.1 draw a label line and label with the letter C to identify the Casparian strip. Alright, so basically I have attached this diagram from the internet over here, which is showing that after the root hair cells comes the epidermis cells, then comes the cortex cells, which are the root cortex cells, then come the endodermis, which contains the Casparian strip, which is basically a waxy strip which prevents the passage of water through the apoplast pathway, and then comes the cells of the pericycle, and then the water enters into the xylem, right? Now basically what is meant by the apoplast pathway and what is meant by the symplast pathway, let us discuss over here. In the apoplast pathway, what basically happens is that the passage of water is basically through the cell wall and the intracellular spaces, right? And what happens in the symplast pathway, that the passage of water occurs through the cytoplasmic extensions which are known as the plasmodesmata, right? And over here in this diagram they are basically showing you the apoplast pathway in which the water is being moved through the cell wall, right? Now in the question, basically let's come back to the question, and they are in the question saying that draw a label line and label with the letter C to identify the Casparian strip. So where is the Casparian strip over here? Basically this is the Casparian strip over here. Alright, so we will label this with the letter C. Alright, so part 2 over here basically says that root hairs measure approximately 5 micrometer in diameter and 500 micrometer in length. Explain how this adapts the root hairs for the absorption of water. We know that the adaptation for the root hair cells is that it provides a large surface area to volume ratio so that the maximum water can be absorbed from the soil, right? So basically these dimensions are helpful in providing the large surface area to volume ratio for the root hair cells to absorb the water more efficiently. Alright, so now let's move on to part 3. Part 3 says that name the pathway for the movement of water shown by the arrow in 1.1. We already discussed that this pathway is basically the apoplast pathway in which the water molecules are moving via the cell wall, right? Cell wall and the intracellular air spaces, right? So this is basically the apoplast pathway as we discussed earlier as well. Now the last part of question number 1, which is part C. Part C over here says that water enters the xylem vessel shown in figure 1.1. On figure 1.1 we can see that water is basically entering into the xylem vessel. Alright. Then they are saying over here that explain how water moves up the xylem vessel to the leaf in a continuous column. This is a very popular question of AS level biology transport in plants, that how basically what is the process of moving the water up from the xylem to the leaves in a continuous column. So we can write its answer in a way that water moves up the xylem via the cohesion tension and the adhesive forces. The two forces are used to move the water up the xylem in a continuous column, that are the basically cohesion tension forces and the adhesive forces. So what happens that the hydrogen bonding between the water molecules allow the strong cohesive forces, right? Cohesion cohesive forces are basically between the two water molecules, right? And the adhesive forces are basically between the water molecule and the hydrophilic part of the lignin in the cell wall. We know that the cell wall of the xylem vessel is basically lignified, right? So adhesive forces of the water molecules are with the lignin of the cell wall, right? So water plus the lignin of the cell wall makes up the adhesive forces for the transport of the water up the xylem in a continuous stream. Now this cohesion and the adhesion creates the tension within the xylem that helps to move the water up in a continuous column. Alright, so basically if we talk about its summary, so the cohesion forces and the adhesion forces you have to talk about in this question, and then you have to say that these two forces combine to make a tension force which helps to move the water up in a continuous column. Alright, so I hope that this question number 1 is clear to all of you. Now let's start with question number 2. Question number 2 is basically from the mitotic cell cycle. Let's read the question. Part A over here says that figure 2.1 shows a cell at one of the main stages of the mitosis in a mitotic cell cycle. Part 1 is saying that name the stage of the mitosis, which is basically shown in figure 2.1. Now if I talk about the stages of the mitosis, so we have basically 4 stages of the mitosis: prophase, metaphase, anaphase and telophase. And if we talk about the main events which are taking place during the metaphase, anaphase, telophase and the prophase, it would be at the metaphase. What happens is that the chromosomes align at the middle of the it aligns at the middle, yeah, here you can say that it aligns at the equator of the cell. In anaphase, from A we can remember that the sister chromatids are pulled apart and the spindle fibers contract. And in the telophase, you can say that in the telophase our nuclear membrane regenerates, nucleolus reappears, right? The nucleolus reappears. And in the prophase, we can say in the prophase what happens is that the nuclear envelope disintegrates, the nuclear envelope disintegrates, the chromosomes become visible, right? So it happens in the prophase. So basically they are over here saying that name the stage of the mitosis shown in figure 2.1. We can clearly see that the chromosomes are aligned at the equator of the cell. So it must be the metaphase. Alright, so the answer to this question is metaphase. Alright, so as I am explaining each and every part in detail, I hope that it is easier for you guys to understand. And if in case of any confusion, you guys can always comment. I'll be there to reply your comments. Let's now move on to part 2 of A. It says that figure 2.2 shows the cell in figure 2.1 at the start of the cytokinesis. Alright, so in the cytokinesis what happens is that the contractile ring is basically formed and eventually the two cells will be formed, right? Complete figure 2.2 to show the daughter chromosomes in each nucleus as it is the mitosis, right? As it is the cell division type is the mitosis. So what will happen that the chromosome number will stay the same. So we are going just simply in this question we are going to draw 4 chromosomes in one cell and 4 chromosomes in the other part of the cell, right? And they will eventually the two opposite membranes will eventually fuse together and 2 separate cells will be formed. Alright, so I hope that this page is clear to all of you. Now let's move on to part B of this question. It says that state the role of telomeres during the DNA replication. Now if you talk about the telomeres, basically telomeres are present at the poles of the sister chromatids. Like these are the telomeres, they are present at the poles of the sister chromatid, and basically these telomeres contain the non-coding sequence of the DNA. Alright, they contain the non-coding sequence of the DNA, and as the cell divides, a part of the telomere is lost. So basically its importance is in preventing the loss of the vital genes which are located over here, right? It prevents the loss of the vital genes. So basically they are asking us in the question that what is the role of the telomeres during the DNA replication. So it allows the cells to divide continuously and prevent the loss of the vital genes. Alright, so this is the most important point you have to write, that it prevents the loss of the vital gene. Right, now let's begin with part C over here. Part C over here is asking us that multiple myeloma is a type of a cancer in the bone marrow. We know that what are the myeloma cells. Basically myeloma cells are the cancerous cells, right? We have studied it in the monoclonal antibody production in the chapter of the immunity, right? So in this question they were saying that multiple myeloma is a type of the cancer in the bone marrow where some of the stem cells starts to produce the abnormal blood cells. One treatment is to collect stem cells from the bone marrow of the person with multiple myeloma. Healthy stem cells are isolated and grown in the laboratory. Then they are saying radiation is then used to destroy all stem cells and cancerous cells in the bone marrow. Then finally large number of healthy stem cells are grown in the laboratory are returned to the bone marrow. Now in the question, the whole story which they have tell is basically to ask you a suggestive question which separates the student of A star and A, basically, right? Because the suggestive questions are the most trickiest questions in the AS as well as A2 level papers, and you have to be very attentive while solving these questions, and these suggestions should be based on the above information and your prior knowledge about this topic, right? So they are saying that suggest the role of the stem cells in this treatment of multiple myeloma. So what happens that why stem cells are important in treating this cancerous multiple myeloma, which is a cancer of the bone marrow. So you have to write the functions, you have to write the role of the stem cells over here. The role of the stem cells is that stem cells divide by the mitosis continuously, right? They are continuously dividing by the mitosis and they differentiate into the specialized cell type, for example the blood cells, right? It helps to replace the cells which are being lost in the cancer. So you can write this and inshallah you will get the number. Over here I have also written that they are undifferentiated cells. The thing which I have written up in the above paragraph, that they are undifferentiated cells, they can divide by mitosis and they can differentiate into other specialized cells which will help to replace the cells which are being destroyed by the cancer. All right, so I hope that this is also clear to all of you. Let's now start. Alright, so now let's move on to question number 3. Question number 3, part A over here says that enzymes are the polymers of the amino acids. Alright, complete the figure 3.1 to show the general structure of an amino acid. So basically the amino acid is having an amine group which is NH2. It is having an amine group which is NH2 on one side, and then it is having a carboxyl group which is on the other side, which is of COOH, C double bond O, OH, and this is basically the side chain, right? This is the side chain and it is a variable side chain. It differs in each amino acid, right? So this is how the structure is to be made. You will write R over here, you will add 2H over here, and then you will complete the carboxyl group over here, right? So I hope that part A is clear. Now let's move on to the part 2. So let's Let's short down this question basically, and let's only read the important points over here. Basically over here they are saying that when the bananas are peeled, the tissue gradually turns brown in the presence of oxygen in the air, and they are over here showing the reaction that how the catechol, which is colorless, oxidizes in the presence of the oxygen with the help of the catechol oxidase and turns brown, which is melanin, right? So catechol and catechol oxidase are present in the banana tissue, and they react with the oxygen, and the catechol oxidase reacts to convert the catechol into the melanin, which is a brown color pigment. Then over here they are saying that as the student investigated how the concentration of catechol oxidase affects the rate of this reaction. Alright, so they are saying that the rate of the reaction R for each concentration of the catechol oxidase used was then calculated using this formula. So basically they have just given you this formula, which is R is equal to 1 divided by time to reach the standard brown color in minutes. And in part 1 of B, they are saying that calculate the rate of the reaction. This is the rate of reaction R. They are saying that when the standard brown color was reached in 2 minutes and 30 seconds. So you just simply have to use this equation, this formula R is equal to 1 divided by time to reach the standard brown color in minutes, where you have to put R is equal to 1 is from this formula, and then this is the time, right? Time is 2 minutes and 30 seconds. Now what we have to be careful over here is that the unit of the rate of reaction that is given in the question is per minute. So you have to make sure that the time you are using is in minutes. So we have to take the time in minutes first. We have to convert the seconds into minutes, and to convert the seconds into minutes we have to divide 30 by 60, right? Because 1 minute has 60 seconds. So the 30 seconds will be equal to how many minutes? So simply we have to divide 30 by 60, right? And then we will plus 2, and then we will put this into the calculator, and we are going to get our answer as 0.4 per minute. The rate of reaction is 0.4 per minute, right? So I hope that this is clear to all of you. The part was quite simple. I think the story was a little longer, but the part was very easy. Now let's move on to part 2. The part 2 over here says that figure 3.3 is a graph showing the results of the investigation. It is a graph between the rate of reaction and the percentage concentration of the catechol oxidase, which is the enzyme in this question, right? And in the graph you can clearly see that as the percentage of the concentration of the enzyme catechol oxidase is being increased, the rate of reaction is also increasing, and you can clearly indicate by it that it is clearly being indicated by the graph, and you can also interpret it from the graph that the enzyme is not the limiting reaction, right? Limiting reagent, right? Enzyme is not limiting in this reaction, right? How? Because as we are increasing the concentration of the enzyme, the rate of reaction is also increasing. Now in the question they are saying that state how the results shown in figure 3.3 show that substrate was in excess at all concentrations of the catechol oxidase tested. Alright, so we know that as the percentage of the catechol oxidase enzyme is increasing, the rate of reaction is also increasing, which means that the enough substrate is present, right? Which means that the enough substrate is present. So how can we interpret it by the graph? That the graph does not reach the plateau phase, right? And if it would have reached the plateau phase, it means that the enzyme is not a limiting reaction but the If it would have reached the plateau phase, it would have indicated that enzyme is not a limiting reaction, but some other factor is limiting. For example, the substrate concentration is limiting, right? Because as the concentration of the enzyme is increasing, the rate of reaction is staying constant, which indicates that the enzyme is not limiting but some other factors, for example the substrate as said in the question, is limiting, right? I hope that this is clear to all of you. Now let's give part C a read. Part C over here says that the student carried out further experiment to investigate how the concentration of the catechol, which is basically the substrate over here of the enzyme catechol oxidase, affects the initial rate of reaction. All the variables were kept constant. Figure 3.4 is a graph showing the effect of the concentration of the catechol on the initial reaction. If we look at the graph over here, so this graph is basically between the initial rate of reaction and the catechol concentration. We can over here see that as we are increasing the concentration of the catechol, the rate of reaction is also increasing initially, and then there is a point at which the graph starts to become plateau, which means that you can say the graph starts to become flat, which means that at this point somewhat over here at this point somewhat you can say that catechol is no longer a limiting reagent, or it is no longer a limiting factor. There may be some other factor now which is limiting, maybe the concentration of the catechol oxidase, which is the enzyme of the catechol, which is discussed over here, may be limiting now, right? So let's now look at the question. They are saying that explain the shape of the curve shown in figure 3.4. So we have already discussed the shape of the curve which is shown, that as the concentration of the catechol increases, the rate of reaction increases as more substrate, so more frequency of the collision per unit time. Basically over here they are saying that as you are increasing the concentration of the catechol, the rate of reaction is also increasing. Why? Because more substrate is there, so more enzyme substrate complexes are formed, and more enzyme product complexes are formed, more product is formed. Alright, so this is what they are telling over here in the answer, that enzyme substrate complex, more enzyme substrate complexes are formed, more product is formed per unit time. So the rate of reaction basically increases, right? The rate of reaction basically is increasing over here. So when the substrate concentration is increased above 1 mole per decimeter cube, like over here you can see that when the concentration increases approximately above the 1 mole per decimeter cube, then the graph starts to become flat or it starts to become plateau, which means that all the active site of the enzymes are filled with the substrate, and substrate is in excess and it is no longer a limiting factor for the reaction of the catechol, right? Enzyme has become limiting now, right? Maybe possible, it is the most possibility that the enzyme may have become limiting because all the active site of the enzymes may have been filled with the increasing concentration of the substrate, right? So I hope that this part is also clear to all of you. Now let's move on to part 2 of B, right? Sorry, part 2 of C. They are saying over here that use the figure 3.4 to calculate the value of the Mitchell's Menten constant, which is Km, and Km is basically the half of Vmax. Km is basically half of the Vmax. If we look at over here, so if we try to calculate it, so we can say that almost the Vmax is coming to be 8, I believe. No, it's coming to be 8.2. Yes, it is coming to be 8.2 because it is coming over here. Yes, if we now draw it, so it is coming to be Alright, so I cannot clearly identify it on the screen, but it's clearly 8.2, which I have also solved over here, that it is 8.2. The Vmax coming is 8.2, and we know that what is basically Km. Km is half of the Vmax, right? And half of the Vmax would be around 4.1, right? It would be 4.1, so it would be somewhere over here, and then the value which will be coming will be around 0.21, right? So the answer for the Km is 0.21, which is the substrate concentration at half Vmax, and half Vmax is 4.1, right? And half Vmax is 4.1. Now let's look at part 3. It says that methyl catechol has a similar shape to catechol. Alright, catechol oxidase can also use methyl catechol as a substrate. Alright, the Km value for the reaction using the methyl catechol as a substrate was found to be much lower than the Km value for the reaction using the catechol as a substrate when their reactions were carried out under the same conditions. They are saying that basically if we use the methyl catechol, let me tell a summary of what they are saying in the question, that if we use the methyl catechol, the Km value is found to be much lower. Alright, it is saying much lower, not higher. It is basically saying much lower. Means that if we use the methyl catechol as a substrate, the Km value is lower, and if we are using catechol as a substrate, the Km value is higher as compared to the methyl catechol. So they are saying state what these Km values indicate about the relationship between the enzymes and the two substrate. So you have to learn this rule, that if there is a lower value of the Km, it means that the affinity of the enzyme and the enzyme for its substrate is much higher, which means that there will be the less amount of substrate will be needed to reach the Vmax, right? The affinity of the enzyme and the substrate will be more, so there will be more, the reaction will be more faster, you can say, and it will reach the Vmax more quickly, right? It will reach the Vmax more quickly in less amount of time, right? And less amount of substrate will also be needed to reach the Vmax. Obviously, if less amount of time is required, so less amount of substrate is required to reach the Vmax. So this is basically what the low value of the Km indicates, that the affinity for the enzyme and substrate is higher, and the high value of the Mitchell's-Menten constant Km indicates that the affinity for the enzyme and substrate is lower, right? Which means that they are not willing to bound to each other, the enzyme and the substrate, right? I hope that this part makes sense to all of you. Now let's move on to the other question, which is question number 4. Question number 4 over here is saying that tuberculosis TB is a major cause of ill health worldwide. Part A is saying state the name of the bacterium that causes the TB in the human. TB is the infectious disease, and the pathogen basically which causes the TB is known as the myobacterium tuberculosis. Alright, this is a bacteria, right? This is a bacteria, myobacterium tuberculosis, which causes the TB in the humans. It is the cause of the infectious disease tuberculosis, right? Now in part B, they are saying that figure 4.1 is a scanning electron micrograph of bacteria that causes TB. Its magnification is given to us. Now they are saying that calculate the actual length of the bacterial cell shown in figure 4.1 along the line XY. Write the formula you will use in the box. Give your answers in micrometers to two significant figures. Alright, so we know that what is the formula for the magnification. The formula for the magnification is the image length divided by the actual length, and the formula which we are going to basically use, we are going to use the formula for the to calculate the actual length, right? So we will make actual length the subject and take magnification in place of the actual length. So actual length will be equal to image length divided by the magnification. We have just rearranged the formula, right? So if we will calculate this length, x and y by the help of the scale, and it is coming to be around 3.4, right? It is coming to be around 3.4 centimeter, and to convert this centimeter to meter, we are going to divide it by 100. So if we divide it by 100, so we are going to get 0.034 meters, right? Then after the meter, we are going to convert it into millimeters, and after millimeters we are going to convert it to micrometers, and we are going to do it by multiplying it by 1000, right? And then the answer which we are going to be getting is 1.62. In place of the magnification, we will use 21,000, and we will use 34,000 in place of the image length, right? So the final answer will be 1.62. I hope that this is clear to all of you. Now let's look at part C. It says that the bacteria are unicellular prokaryotic cells with a diameter of 1.5 micrometers. Now in the next part, they were saying that state two other structural features that would identify cell as a prokaryotic cell. So they are saying that which structures would identify the cell as the prokaryotic cell. So it would be a circular DNA. We know that circular DNA such as the plasmids are the identification of the prokaryotic cell. They are found in the prokaryotic cell. We all know that the cell wall of the bacteria is made up of peptidoglycan. So this can also be an identification also that the prokaryotic cells or the bacterial cells does not contain any membrane bound organelles. So non-membrane bound organelles are only found in the only the non-membrane bound organelles are found in the prokaryotic cell. So this can also be used as an identification. Also in case of the ribosomes, only the 70S ribosomes are present in the prokaryotic cells. So we can also raise this point as well. Alright, so in part D, they are saying that the World Health Organization WHO published a data in which they estimated the number of deaths from TB as well as from HIV in 2018. All deaths of the people from TB who were infected with HIV were also counted as death of the people with HIV. Alright, so basically we have to understand this statement that what they are saying that all deaths of people from the TB were infected with HIV were also counted as death of people with HIV, right? Look over here, they are showing you that the deaths of the people from TB which were not infected by the HIV, and here they are showing the death of the people from TB which were affected by the HIV, which are smaller in proportion than not affected by the HIV, right? And then that of the people with HIV AIDS is showing the not infected with this proportion is affected from HIV but not from TB, right? And this is affected from TB, right? So now they are saying in the question basically that a student used the data from figure 4.2 to predict the measures to control the spread of HIV will decrease the number of deaths from TB. So basically they are saying that if we control the spread of HIV, so it will decrease the number of deaths from TB, right? Number of deaths from TB will be reduced if you control the HIV, they are saying. Now they are further saying in the question that discuss whether the data in figure 4.2 supports this prediction or not. Alright, so now let's read the answer and check whether it is being supported by the data or not. Majority of the deaths from the TB are in the people not infected by HIV, right? Most of the death of the TB are in the people which are not affected by the HIV. So it is one of the statement which is not supporting this prediction, right? This is not supporting the prediction, So obviously the prediction is not being supported by the data given, right? The measures to control the HIV spread may not prevent the spread of the TB as the mode of transmission is different. Obviously we know that the spread of the TB is by the water droplets of the cough, right? Water droplets of the cough. Basically, the mode of transmission is coughing, right? When the water droplets come in contact with the other person, so then the spread of the TB is there, right? And if we talk about the HIV, so HIV is basically spread through infected blood needles, right? Infected needles, serums, right? So basically, data needed to know that whether HIV increases the risk of dying with TB or not. So we need further data for this as well to basically understand even more that whether the HIV will increase the risk of dying with TB or not. We need more data for this, right? Otherwise, if we talk about the conclusion, so it is basically not supporting, right? So I hope that this question is clear to all of you. Now let's give part E a read. Part E says that in healthy people the number of T helper cell ranges from 500 to 1200. In untreated people which are basically infected by HIV, the number of T helper cells can decrease to below 200 cells per centimeter cube of the blood, right? Now we have to understand the function of the T helper cells. Basically, T helper cells are considered to be the king of the immune system. They are the king of the immune system. They release the chemicals which are known as cytokines, which activates the other cells of the immune system to carry out an immune response. In the when the T helper cells get the stimulation that in foreign antigen is there, so they release the cytokines which activate the other cells of the immune system, right? So they are basically saying that we have to in this question explain that how a low number of T helper cells makes it more likely that untreated people infected with HIV will die if they are also infected with TB. We know that in the HIV the T helper cells will be less, right? So when the T helper cells will be less, so less T helper cells will cause less cytokines to be released, right? So the pathogen or the foreign antigen cannot be very quickly recognized. It will be slowly recognized, right? So there will be poor stimulation of the macrophages and the B lymphocyte. There will be poor stimulation of the macrophages and the B lymphocytes, and we know that B lymphocytes differentiate into plasma cells, which makes the antibodies, right? So there will be poor stimulation, so the immune response will be late, right? Plasma cells are made late. B lymphocytes, if B lymphocytes will be stimulated late, so plasma cells will be made late, and if plasma cells will be made late, then the antibodies will be made late, right? And the infection may have already been spread among the body. Question number 5 is from the transport in humans chapter. It says that control of heartbeat is myogenic. This means that the electrical activity controlling the rhythm of the regular heartbeats begin within the muscle itself, within the heart muscle itself. Atrial fibrillation AF is an abnormal heart rhythm that causes rapid and irregular contractions of the atria. Untreated cases of AF can lead to stroke. Now in part A they are saying that a stroke is caused by when a small blood clot often forming in the left atrium is carried by the blood to the brain, where it blocks a small artery and leads to the brain damage, right? Now in part 1 they are saying list all of the structures through which a blood clot in the left atrium must have traveled to reach the blood vessel supplying the brain. The structure must have been listed in the correct sequence. So basically now in this question they are saying that after the left atrium, which pathway the blood will take to reach the brain. सही है. So first of all, after the left atrium we know there will come a bicuspid valve, right? And then there will come the left ventricle. Then there will come a semilunar valve, and then it will come the aorta, and aorta is the main artery which supplies the oxygenated blood to the brain and the other parts of the body as well. Now in part 2 they are saying that explain why blocking the small artery in the brain leads to the brain damage. We know that if the artery, we know the basic function of the artery is to supply a specific part with the oxygenated blood, right? And if that small artery will be blocked, right, and if that small artery will be blocked, so there will be the lack of oxygen, uh, there will be the lack of oxygen which will be provided to the brain cells, so the brain cells would not respire, they cannot respire, right, and they may die, right. So this can be the answer of part 2. Now let's read part B over here. Part B over here says that a common cause of AF is when a small group of muscle cells in the wall of the left atrium starts to send out electrical impulses to the surrounding heart muscle cells. Explain how the control of the heartbeat by the sinoatrial node can be disrupted by AF, resulting in rapid and irregular atrial contractions. We know that sinoatrial node is basically the pacemaker, right? It is where the heartbeat is initiated, right? The electrical impulses are basically initiated from the sinoatrial node and then they go to the AV node, right? So they are saying that how the AF will result in the rapid and irregular atrial contractions. Rapid atrial contractions would mean that extra contraction, right, extra contraction of the extra contraction of the muscles of the heart, right, because of the extra impulses produced as a result of AF, right. And irregular atrial contractions would occur because there would be a loss of the sinoatrial node control, right. We know that first the sinoatrial node comes, then the AV node comes, and there is a short delay after the sinoatrial node sends the impulse to the AV node, right. AV node conducts later, right, so that the atria may fully contract. So there will be a loss of the SA control if there will be the irregular atrial contractions. And they are specifically talking about the left atrium over here. So we know that the sinoatrial node is present in the right atrium. So as the right atrium and left atrium contracts together, right, so that's why right sinoatrial node produces and left atrium will also contract, right, because both the atriums contract together. So this can be the answer of part B over here. Now let's move on to part C over here. Part C over here is basically about the transport of carbon dioxide and oxygen. There in the figure they are showing that the figure for 5.1 is a diagram representing the exchange of oxygen and carbon dioxide between a red blood cell in the capillary and in the respiring cell. This is the respiring cell over here, and this is the capillary wall over here, and over here and contained within the capillary are the blood, and the blood contains the red blood cell, right. Some of the reactions that take place in the red blood cell are shown. Now they are saying that identify the enzyme X and the molecule Y in this diagram. So this is a very popular reaction that when the carbon dioxide basically combines with the water, it forms the carbonic acid, right. This is the carbonic acid H2CO3, which further breaks down into HCO3 and H positive, and as the H positive goes out into the extracellular space basically, so the chloride ion enters into the red blood cell to maintain the electric balance of the cell. The enzyme which is used over here, in the reaction of carbon dioxide with water is basically the carbonic anhydrase enzyme, right. So X is basically the carbonic anhydrase enzyme, and when the oxygenated hemoglobin will combine with H positive ion, so it will form the hemoglobinic acid or hemoglobin and oxygen. Basically it is showing the dissociation of the oxygenated hemoglobin, right. So it will form the hemoglobin, or we can also say hemoglobinic acid. Now let's have a look at part 2 over here. Part 2 over here says that the hydrogen carbonate ion shown in figure 5.1 leave the red blood cells and are replaced by the chloride ion. As I told you earlier over here that hydrogen carbonate ions leave and the chloride ions enter. Now they are asking that why is it necessary for the chloride ions to enter the red blood cells as the hydrogen carbonate ions leave. So basically, why is it important to maintain the electrical neutrality of the cell? Obviously it maintains the electrical neutrality of the cell. Chloride ion enters the red blood cells to replace the hydrogen carbonate ions, the negative ions leaving the cell, right? So it will balance the electrical neutrality or electrical charges of the cell, right? In part D, they are saying that identify the aqueous environment labeled Z in figure 5.1 that surrounds the respiring cell. So basically this, they are saying that what is this environment basically. So we know that the fluid which leaks out of the walls of the capillaries from the blood is known as the tissue fluid, right? So its answer will be tissue fluid. In part E, they are saying oxygen and carbon dioxide are also exchanged between the blood capillaries and the alveoli in the lungs. The gas exchange system has specialized cells to prevent harmful microscopic particles that are present in inhaled air from reaching the alveoli. The particles are associated with many respiratory diseases. Explain how specialized cells in the gas exchange system prevents the harmful microscopic particles from reaching the alveoli. So basically over here they are talking about the adaptations of the respiratory epithelium, right? If we talk about in short, so they are talking about the adaptations of the respiratory epithelium, and in the respiratory epithelium we have the goblet cells and we have the ciliated epithelial cells. The reason for having the goblet cells is that these goblet cells release the mucus, and what is that function of the mucus? Which helps to trap the bacteria and the dust particles, right? And then the mucus goes up to the pharynx to be swallowed, right? And then the ciliated epithelium, they have the cilia that have the beating movement, right? Like for example, this is the epithelial cell and they are the cilia over here, so they have the beating movement, right? They move up and down. So what happens because of this beating movement, that it moves the mucus up to the pharynx to be swallowed, right? Ciliated epithelial cells also trap the bacteria and the dust particles, but its main function is that the beating movement basically moves the mucus up to the pharynx to be swallowed, right? So I hope that this question is clear to all of you. Now let's move on to the last question of this paper, which is February March 2022. Question number 6. Question number 6 over here says that figure 6.1 is a diagram showing the structure of a part of a DNA molecule. And as you know about a DNA molecule, in DNA molecule the sugar which is being used is a deoxyribose sugar, which is a 5-carbon sugar, right? It is a 5-carbon sugar, right? So this is basically the structure of the DNA molecule over here. These are the hydrogen bonds, these are the nitrogenous bases, this is the phosphodiester bond, right? Now let's look at the question, basically what they are asking us in the question. So in part A1, they are saying that identify the structure E and the structure F. Alright, so the structure E is definitely the sugar, and we know that it is a deoxyribose sugar. Why? Because it is a DNA molecule, right? And then because they are talking about a DNA molecule, we know a rule about base pairing, that adenine will pair with thymine with 2 hydrogen bonds, right? And then guanine will pair with the cytosine in the presence of the 3 hydrogen bonds, right? So it is a very good way to identify. Also we have to tell about F, right? So F is over here. So what can be this? It is a single pyramid, it is a double pyramid. The structures will double pyramid or the double ring structures are known as the purine, right? And the single ring structures are known as the pyrimidine, right? So if it is binding with the uh, if this structure, which is the pyrimidine, it is binding with the purine and it is having 3 hydrogen bonds, it means that it is a cytosine, right? So this is how we are going to conclude it in the exam as well. We have to keep our mind very active while solving these type of questions. Now in part 2 they are saying that on figure 6.1 draw a circle around one nucleotide. So this is one nucleotide. A nucleotide contains a nitrogenous base, a phosphate group and a sugar, right? It can be the ribose sugar or it can be a deoxyribose sugar depending on the molecule, whether it's DNA or RNA. Now they are saying in part 3 that state the name of the covalent bond that links the two nucleotides together. I already told you guys that this bond which is linking the two nucleotides together is the phosphodiester bond. It is a very famous bond which you guys study in the nucleic acid chapter, right? Now let's look at part B. Part B over here says that figure 6.2 shows the RNA based sequence of a short length of a primary transcript. It is a RNA based sequence of a this is the RNA sequence of a short length of a primary transcript, right? This is the RNA sequence and they are saying that complete by writing the DNA based sequence. So basically you can say that over here maybe transcription would have happened, right, from where the mRNA is made from DNA, right? So they are saying that we have to fill in these blocks. Which blocks we have to fill in? These blocks. So we know that A binds with and G binds with C and A binds with U, right, in case of the RNA molecule. So wherever there is A, you are going to write, wherever there is U, you are going to write A, right? And wherever there is A, you are going to write T over here, because in case of the DNA molecules T is used, and if there is U on the RNA molecule, then A is used. Otherwise, if there is A on the RNA molecule, then T will be used, because you are going from the mRNA to the DNA, right? So it will be GAT, then A is given, so T and U is given, so it will come A, right? CGA will be there because G bonds with the C, right? And then G bonds with the C again, and A bonds with the U in case of the RNA structure. And then GGU would be complementary to CCA, right? So I hope that this makes sense and it is clear to all of you. Now in part C, they are over here saying that in eukaryotic cells, the primary transcript is modified to form mRNA. Explain how the primary transcript is modified to form mRNA. Basically, the primary transcript which they are talking about over here is the pre-mRNA, right? And then that pre-mRNA undergoes the post-transcriptional modification and then becomes the mature mRNA, right? Which is further translated into the protein, right? So now they are saying that explain how the primary transcript is modified to form the mRNA. How it is modified, basically they are saying. Now it can be modified in number one way, that splicing can happen. Splicing, what is splicing? Basically in the splicing what happens, that the introns, which are the non-coding regions, on the mRNA, pre-mRNA are removed, and the exons, which are the coding regions, are joined together, right? And exons are added together, right? Introns are removed, which are the non-coding sequences, they are removed, and the exons are added together. So the splicing happens, right? And then there is the 5' capping, right? At the 5' end, what happens is at the 5' end of the mRNA, pre-mRNA, at the 5' end, there is basically a 5' capping, right? And in the 5 prime capping, basically what happens is that a modified guanine is a nucleotide base, right? So a modified guanine nucleotide is basically added to the 5 prime end of the pre-mRNA, which is known as the 5 prime capping, right? और इसका हमारे पास the benefit of it is that it protects the mRNA from the degradation, right? इसका the addition of the guanine, which is the 5 prime capping, and the addition of the poly A tail to the 3 prime end, both of the modification have a very similar function, that it protects the mRNA from degradation, right? It protects the mRNA from the degradation. Now basically what is the now let's discuss that what is the addition of the poly A tail. Basically poly A tail हमारे पास क्या होती है, if I underline it over here, if I highlight it over here, so poly A tail basically it is a long chain of adenine nucleotide. It is a long chain of adenine nucleotide, right? Nucleotide, it is a long chain of adenine nucleotide, and it is added to the 3 prime end of the mRNA molecule, and it protects the mRNA molecule from the degradation. I hope that this part is clear to all of you. Now let's move on to the last part, which is the part D over here. Part D over here says that the mRNA strand is translated at the ribosome to form a polypeptide. Okay, we know the process of translation where mRNA is converted into the polypeptide chain with the help of the tRNA. Alright, now they are saying describe how the process of translation results in the formation of the polypeptide. Alright, so I have an image for the reference which I have taken from the internet. Over here you can see that the mRNA molecule is attached to the small subunit of the ribosome. Over here it is a small subunit of the ribosome, it is the large subunit of the ribosome. We know that the ribosome is made up of 2 subunits, right? A small subunit is there and a large subunit is there. So the mRNA molecule goes and binds to the small unit of the ribosome, right? And then what happens is that the mRNA is basically having the codons, and the tRNA molecule, this is a tRNA molecule, and it is having the anticodons. Alright, so the transverse RNA, let me erase the extra stuff. So that transverse RNA, which is complementary to the bases, will be activated, and they will carry a particular amino acid to the mRNA, and then the polypeptide chain will be formed in a sequence, right? The polypeptide chain will be formed, and then more will come, the more tRNA will come until and unless the whole mRNA has been translated, right? So I hope that the process is clear to all of you. Now let me read the answer, that how you have to write it in your examination. Now the way you will write it in your examination is that the mRNA will come and attach itself to the small subunit of the ribosome, as we saw over here, right? And then what is going to happen, that the 2 codons are exposed at a time. Alright, codons are present on the mRNA. It is not so much specific to write. It's your choice if you want to write this statement or not. The 2 tRNA molecules brings amino acid at a time. tRNA brings specific amino acid to a ribosome. It binds to a specific binding site on the ribosome. Then the anticodon binds to the complementary codons, right? You can see over here the binding is between the complementary codons and anticodons over here, right? The G is binding with C, the A is binding with U. Then what will happen, that the both tRNA molecules hold the amino acids close together just like this. For the peptide bond formation between the adjacent amino acid, this process is repeated until the stop codon is reached in the mRNA, right? The tRNA molecules can be reused. After it drops the amino acid at the side of the polypeptide synthesis, right? Alright, so this is all about the February March 2022 paper 2-2. I hope that each and every question is clear to all of you. And in case you are having any doubt in the parts of the question above, you can always say it in the comment section below. I will be there to answer it, inshallah. If you guys enjoyed solving the past paper with me, then do consider subscribing this channel. Your prayers and your support means a lot to me. Thank you so much everyone for joining in. I hope that they were beneficial to all of the students. Also, do tell me in the comment section below that which paper you want me to solve next, so that I may remember your recommendation next time I'm making the video. Allah Hafiz everyone. Take care. Have a nice day.